So Magdalena did a PhD in the Department of Botany in Prague, and then now she's a Marie Skłodowska-Curie Fellow with Katie Peichel in Bern. She's done a bunch of work on parallel and convergent evolution and adaptation in Arabidopsis, including to understand what happens after genome duplication, to make for a nice transition.
Thank you, and I'm very, very honored to be here, so thank you for the invitation. And I will directly start with a question. Do you think that we can predict adaptation? It's definitely interesting from evolutionary biology perspective because it informs how much determinism or stochastic events contribute to evolution. And in theory, it might have also some practical implications. For example, it would be very cool to predict the evolution of antibiotic resistance, pesticides resistance, or what happens after human-induced environmental change. To make this introduction more fun, I decided to generate these pictures using artificial intelligence. I really like the cubistic set Earth. And I like to look into this question of predictability of adaptation by studying repeatability of adaptation in nature. So what I mean by repeated adaptation, by this word I refer to independent evolution and adaptation under the same selection pressure in different lineages. And I am a plant biologist, but I recently moved to a fish lab. So here I prepared a 3-spine stickleback example, which is a marine fish, but which repeatedly adapted into the freshwater environments every time that you see the blue dot on the Earth. And I'm interested in the repeated adaptation at the genetic level, asking how frequently organisms use the same genes to adapt to the certain environmental pressure. And this could be either due to the same mutation in the same gene, or it could be also different mutations in the same or different parts of the genes. So this would be one scheme how to represent this, and then I would call this repeated use of gene in the adaptation a gene reuse. And if you don't enjoy schemes so much, I can also show you an example. Again, in the stickleback case, you might know that anytime it colonized freshwater, it increased the frequency of the EDA allele of the EDA gene, which helped into the adaptation. So this particular gene would be case of gene reuse. It, of course, also used several genes which were not reused. So if we assume that this repeatability in nature also to some extent informs about the predictability, we might maybe think that we just need to look into the degree of gene reuse between different lineages, and that should roughly tell us to which extent we can assume that the adaptation would be predictable. But it's of course not this simple. Nature is very diverse, and if we look for the specific cases of repeated adaptation, we see very different proportion of gene reuse. Here I return back to my safe plant kingdom, and I prepared 2 examples. So there is adaptation to Arctic environment, which you can imagine it could be quite challenging, in 2 species called Draba and Cochlaria. It was studied on the genetic level, and they found that these 2 species reuse exactly 2 genes to adapt to that environment. On the other side, there was a study of repeated adaptation to toxic serpentine cells in different Arabidopsis lineages, and there was around 30 reused genes per lineage, and given that the total number of genes which were identified to contribute to these adaptations could be around 100, this would be like 30%, so quite high gene reuse. And I should also say these are all Brassicaceae species, which have around 30,000 genes described, so the numbers might be comparable. So now my question is why? Why we see this variability in gene reuse? What determines the fraction of genes which would be used repeatedly between these different instances of adaptation. And here I give you a few moments to think about it. You can also use the example which I showed here to think, to come up with some like explanation. I believe there is multiple explanations, but during this talk, I would like to offer you some empirical evidence for one of the factors which might be behind this variability, and that is a divergence time. So here, if I put the divergence between the species studied as another information, the Draba and Cochlaria were estimated to be around 20 million years diverged, whereas the different Arabidopsis lineages were only few thousand generations diverged. So if we put it into the scheme, we might propose or expect that with increasing divergence between the repeatedly adapting lineages, the degree of gene reuse in their adaptation would decrease. And there is actually already slightly different evidence which might be in this line, and that is first a meta-analysis which showed that the degree of phenotypic convergence is decreasing with increasing times, separating the taxa, and also the degree of gene reuse underlying specific traits, so not adaptation, but like one specific trait in the organism, is decreasing with increasing divergence between the taxa. And my questions regarding the expectations are first, can we find empirical support for this relationship by studying cases of repeated adaptations in nature? And second, and that's the interesting part, if yes, can we find underlying mechanism behind this relationship? And today I prepared 3 case studies or indications, which I did. First would be at a short time scale, second at the intermediate, and the last one is actually a meta-analysis of existing literature across all scales. So what type of setup do we need to perform such analysis? So of course we need multiple lineages, and these lineages should differ in their divergence time, the neutral divergence time, which is shown there by the red and yellow times. And all of these lineages should also contain populations which adapted to certain comparable selection pressure in a similar time point in the past. So what is different between these cases is the neutral divergence, but the time since adaptation optimally should be similar. And I have 2 model systems to study it, or model selection pressures. First is high alpine environment, so above a tree line, and second is whole genome duplication. The second one might be a bit more tricky to imagine, so I will explain a bit. Whole genome duplication happens when the organism doubles their chromosomes, and is usually considered either evolutionary death end or even evolutionary opportunity. But I don't care here about any of these considerations, but I consider it a strong inner selection pressure. Because if organism has twice as many chromosomes, perhaps they have bigger nucleus and also the whole cell size, they somehow have to deal with it before the lineage is established. So I just take it as a case of inner environment which is driving adaptation. And then, having all of these instances, we basically go and sample multiple lineages, always both from the ancestral environment, which in our case is either foothill or the diploid lineages, and then the derived challenging environment, which would be the alpine or the tetraploid ones. And then we use these contrasting environments, so foothill, alpine, and we used sets of methods called selection scans, where we identify what we call candidate gene sets. So sets of genes which might likely contribute to that adaptation. We do it within each of the lineages, and then we overlap them, and what gets overlapped is what we call a gene reuse. And then if we want to calculate the degree of gene reuse genome-wide, we would just do the ratio of the overlap of the genes to their union. Okay, so let's have a look into the real natural cases. So I will start with the alpine adaptation in the Arabidopsis species. You might know the famous Arabidopsis thaliana, which is the model system in plant biology, but here I use I'm not using directly that species, but its wild relatives, called Arabidopsis arenosa and Arabidopsis halleri. These are predominantly foothill species with relatively tall, white-flowered phenotypes, but occasionally they are able to colonize a high alpine environment. In that case, they evolve a short, pinkish phenotype with thick wax layers and many other different traits. And we identified at least 7 instances of this alpine adaptation in the 2 species. I wonder, pointer, great. Great. 5 are in Arabidopsis arenosa. Here on the map, they start with A, and 2 are in Arabidopsis halleri, starting with H. And what is important for our questions is that these instances of adaptation differ in their divergence time and they vary from very recently diverged lineages up to the different species level, which is roughly 1 million generations ago. And because we have 7 lineages, that makes it very crazy to overlap everything with each other. So this is a Venn diagram of if you try to overlap 7 entities. We decided to, instead of overlapping everything, to work with the 21 possible pairwise comparisons. And if we looked for the degree of gene reuse among these 21 pairs and their divergence, we identified this quite clear and also significant decreasing trend. So that is a nice empirical evidence for this divergence dependency of gene reuse. To integrate it a bit with ecology, we also had a look into these reused genes. It was overall a set of 151 genes. And if we were able to retrieve the annotations, they mostly related to the logical, ecological stressors which are experienced by plants in an alpine environment, like low temperatures, high radiation, short summer seasons, and altered ion composition or the spectra of herbivores and pathogens. So we have the relationship now, and the interesting question is, what's the underlying mechanism of this divergence dependency? And based on the literature, we proposed it could be a reduction in allele sharing. So what do I mean by allele sharing? It occurs when the 2 lineages don't have independent mutations, but they share their variability from either a common ancestor, which I try to visualize here, or as a mutation which originated in one of the lineages, but then got introgressed into the other one. We can imagine like a seed flying into another mountain range. And given that these shared alleles, by definition, have to occur in the very same gene, and also that they tend to be of higher frequency than the alternative, which is the independent mutations in the genes, they are also likely to be contributing to the gene reuse more than the independent mutations. So if you have more allele sharing, you should also have more gene reuse. But as lineages diverge, their chances to share alleles are decreasing, first because they accumulate hybridization barriers, and also just due to the random draft after the speciation. They are just losing everything which they had in common originally. until they no longer share any alleles. And if we sum up this effect for each gene genome-wide, this decreasing allele sharing can also lead to the decreasing degree of gene reuse. So that would be our expectation. This was the set of genes for which we were able to use modeling to find out what was the source of their alleles, if it was adaptive introgression, if it was standing variation, or if they were from de novo mutations. And once we calculated it, we again got this decreasing trend in which the allele sharing for these underlying these genes was decreasing with increasing divergence. And then we were able to statistically show that most of this relationship was actually explained by the decreasing allele sharing. So to wrap up this first case study at the shallow divergence levels, we were able to identify support for decreasing gene reuse with increasing divergence, and we mostly explain it by reduction in allele sharing. So this brings this little check mark for Arabidopsis here. Now let's have a look at the intermediate timescales where we studied the adaptation to whole genome duplication. So as I said, the polyploid, in our case tetraploid, plants have more chromosomes, so they have to figure out their cell cycle, their misregulation of transcription, and also the things related to the cell size. And here are our three superstars, polyploid superstars. Arabidopsis arenusa you already know as the alpine adaptation plant, but it is also diploid and tetraploid. Then Arabidopsis lyrata and Cardamine amara. And because we wanted to get a bit outside of this allele sharing area, we selected a species which are more diverged. So these two are still sister species, but they are around 1 million years diverged. And then Cardamine amara is really diverged by around 17 million years ago. And I should say that these all share distribution ranges in the Central Europe, so in that respect they should be comparable, and they are all outcrossing, which means they are genetically diverse. So it's easy to identify the signatures of selection even in the polyploid species. And these were the results. So while these two sister species had relatively high gene reuse, and it was very significant, they shared only limited number of genes, and that was non-significant overlap with the more distantly related Cardamine amara. So if we put this into now very simple graph, this would again point towards the decreasing divergence dependency. So why is it so? And again, actually, this high sharing here was due to the allele reuse. And the reason is that while these two species are well reproductively isolated at the diploid level, this is a structure plot of the diploid populations of both species and then their tetraploid lineages, tetraploid populations. And you can see that they are well separated at the diploid level, but then due to some polyploid magic, which I won't be speaking about now, they are able to break the reproductive isolation, the post-zygotic reproductive isolation, which they have between each other at the tetraploid level, and they can exchange alleles, which they later used for the adaptation to whole genome duplication. It's actually a very funny case in which the cause for the adaptation is also providing the means to adapt to it. So it's almost a circular thing. While Cardamine amara, which is really diverged, is not able to share these alleles with these two, and then the genetic architecture of that adaptation becomes more independent. And finally, we asked if this could be compensated, this lack of gene reuse between Cardamine and or limitation of gene reuse between Cardamine and Arabidopsis could be compensated by functional repeatability. And we predicted protein-protein interactions between the candidate gene sets found in the Arabidopsis and in Cardamine. And we actually found that they very often cluster in the same protein-protein interaction network. So this gives a support that these genes are different, but they contribute to very similar functional process. And these networks were related to first meiosis. This is a meiotic network, and it's a DNA repair part of meiosis. And also ion transport, which could be related if you increase your cell size, you also change your cell surface to volume ratio, and the proportion of transporters no longer fits what's optimal for the plant, so they might also change the ion transport for that. Okay, so again, we identified an empirical support for the negative divergence dependency, but again, this was mostly probably due to the reduction of allele sharing. We were not able to think of like further mechanisms behind that. So another check mark here. And this was basically when I finished my PhD in 2021. And right afterwards, we started to see similar trends appearing in other case studies. So this would be a thimemastic insect. This X-fold here is basically a measure of gene reuse, and again, a negative divergence dependency. Then we found similar in stickleback and in the heliconius butterfly. And in the last one, it was even attributed to the reduction of allele sharing. So this motivated me after I moved to Katie Peichel's lab to start a meta-analysis of basically published divergence scales in repeated adaptation across literature to see if we can find a support across the full divergence scale for this relationship, and more importantly, if we can understand something more about the mechanisms. Do I still have a time? Okay, good. Good. Oh, that's also good. 4 minutes is perfect. What was our expectation? So as I said, I hope I said it, the allele sharing can't go forever, because as long as you speciate, you will slowly get rid of the shared variability. So we expected there would be some limit after which this wouldn't be mechanism underlying the divergence dependency. But then there is quite some knowledge in the literature that both genome itself and also its architecture and functions of orthologous genes are diversifying with increasing divergence. Here I prepared a little scheme. So if these are the two orthologs of the two different lineages, they would initially perform very similar function, but as these lineages diverge, they might perform a new function, or perhaps be lost or duplicated, and then the duplicate takes the function. So under this scenario, you don't expect any gene reuse anymore. And the divergence dependency of this process is quite well monitored in the literature. So I expected this would naturally lead to the continuation in the decrease of gene reuse with increasing divergence. And what were the data sets? So in total, we were able to retrieve 28 studies which studied repeated adaptation on the genomic level and had a divergence scale in between among the different lineages. And this scheme is showing one arrow for one of each of the case studies. And if it's the green triangle down or the green arrow down, it shows our predicted negative divergence dependency of gene reuse. The black ones are just not showing any relationship, and there are even few which show the opposite one, so more gene reuse among more diverged populations. And I think we can basically split this divergence scale into two halves. First one is these more shallower parts in which we see quite some, and it's also statistically significant support for the negative divergence dependency. But then at deeper divergence levels, there isn't any actually support for any of these scenarios. They are roughly the same proportion. So basically, what I can wrap up here is that we have quite some evidence in the literature for why the gene reuse should decrease with divergence, but we miss a support that it is actually so. So to conclude, it seems that divergence matters, but it's very matters for the degree of repeatability of adaptation, but this is very much dependent on to which part of the divergence scale you look at. And if we should put this into bigger picture about the predictability, which is the framing of this presentation, it could be that adaptation might be to some extent a little bit predictable, but it's definitely more so among very closely related lineages, and it probably becomes less and less evident as they diverge. And then, regarding the absence of the trend across this in this meta-analysis, what we actually missed there, and this divergence scale is a logarithmic scale. So we missed studies which would really span a huge divergences. So studying the same selection pressure and the repeatability of it across different kingdoms, but starting at very related populations and then going further and further. The question is whether we can find a selection stressor which would affect enough conserved genetic pathways that we can even trace them across all of these divergences. My guess is that it should be possible with adaptation to whole genome duplication, because as it is inner stressor, it somehow and affects things like meiosis, which is shared across eukaryotes. We might be able to identify the shared genetic basis, or at least expect it, but I don't think if there is more of such selection pressure, so I would be happy to maybe discuss it later if you have any ideas. And it would be also very nice to have clear knowledge about what specific mechanisms can be behind this relationship, and if we can time them, because like timing functional diversification could be very challenging. And with this, I would like to thank you for your attention, and both of the group I was part of in the recent years, which is the Planck Ecological Genomics group of Philip Collège in Prague, where I did my PhD, and now the Evolutionary Ecology group of Katie Peichel in Bern. They are both super friendly groups, and I'm very lucky about the people I can work with. And finally, regarding this meta-analysis, we are currently writing a review with Katie Peichel, and it's still very much under process, so if you would be willing to discuss it with me, have additional ideas, or even read a manuscript for us, that would be super, super nice, and you can catch me around, and I would be very, very grateful for any feedback. I think this is a perfect place where to discuss it. Okay, thank you.